Okay, everyone. Hello. Today, I'm going to talk to you about genetic challenges and certification. This is with respect to St. Augustine grass. Before we get into that, let's all remember that we have a long history of St. Augustine grass use in Florida. In fact, some of the cultivars that are still in use today have been around a really long time. And in fact, Bitter Blue has been around for almost 90 years. Floratam has been around for close to 50 years now, and we're very fortunate to have two new varieties recently enter the market, those being Provista and Citra Blue. Having more options in the market is good for diversity and helps the market to grow and expand. So on with genetic challenges. St. Augustine grass is produced as sod. There's no seed that can be purchased or available. And if we look at these sod fields of St. Augustine grass, we see these ribbons that are left because St. Augustine grass doesn't have underground stems. It recovers or regenerates from stolons. And this is very simply an example of vegetative or clonal reproduction. And so every plant in a sod field of a particular cultivar should be genetically identical due to vegetative reproduction. So in that context, why do we have genetic challenges? How, if a field is clonally reproduced, does contamination occur? So I like to focus on the right part of the slide here with the seed heads. And this upper picture is a typical St. Augustine grass seed head that we should all, I assume, are familiar with, have seen. And St. Augustine grass doesn't put up a big showy seed head like Bahia grass, but it does produce seed heads down near the canopy, sometimes within the canopy, and you may not see them. So this entire structure is called a spike. And then these structures here that you may think of as a seed, this is a spikelet. Within that are florets, the floral structures of a grass seed head. And then the close-up down below is a seed head that is in flower. The florets are in bloom. And here we have the male flower or the anther and the female flower or the stigma. It's this feathery structure here. So the anthers dehisce pollen, which fall on the stigmas and fertilize the plant. If this happens to be a fertile St. Augustine grass cultivar, a seed could form. Within St. Augustine grass, we'll have two broad morphotypes that I'll discuss. You could get much more specific than this, but I'm going to use the terms diploids and polyploids. And these have to do with chromosome numbers. You may be familiar with terms such as dwarf or semi-dwarfs that I'm using to associate with diploids. Now, if we look at the cultivars down here, we don't tend to think of Raleigh as a dwarf or semi-dwarf, but it is a diploid, so it's included in here. The point is that diploids tend to be fertile and produce seed, and the non-diploids, we'll just lump them into polyploids, and that would be Bitter Blue, Floratam, Provista, Citra Blue. They tend to have poor fertility or low or no seed set. So we're going to explain this a little bit more. So diploids have a total of 18 chromosomes. Again, the term diploid is a reference to the number of sets of chromosomes. We have a total of 18 in diploid St. Augustine grass. We don't have 18 different chromosomes. We have 9 different chromosomes and 2 sets of those. So I'm going to show one set of 9 here, and I'm just very simply using numbers to represent the different chromosomes. We could make this much at several different levels of advancement, but we're going to keep it very straightforward. And now we have a second set of 9 chromosomes. So what we have here are 2 sets of chromosome 1, 2 sets of chromosome 2, so on and so forth, so that we have 2 sets of 9. And so we have, in terms of cell division or meiosis to produce gametes, the 2 chromosome 1s, which are identical chromosomes, except that they may have different forms of the genes. They have the same genes, but maybe different forms. So that's why they're different colors, to help you realize they may have different forms of the same genes. And so in cell division, the two chromosome 1s would pair and then they would divide for gamete production. The two chromosome 2s would pair and divide for gamete production, and so on and so forth. And the way these things would separate are at random from one another. So that means that a gamete or an egg or pollen cell will only have one set of 9. And a gamete will have random combinations of the parent's chromosome. And so we're showing a potential gamete that could have formed from the female flower and a potential gamete that could have formed from the male flower. And you see that these are essential mosaics of the parental combination, but each gamete has a set of 9, one set of 9. Gametes unite to form a new diploid St. Augustine plant with 18 chromosomes. So because this process works very smoothly in diploids, they are fertile. If we think about the polyploids now, specifically a triploid, will have 27 chromosomes or 3 sets of 9. One of the points to make back here with diploids is that we have an even number of sets. So those sets can pair up and divide very easily. If we have an odd number of sets, these can't pair up and divide evenly. So you can imagine that gamete formation will be a significant struggle in a triploid. And so fertility of a triploid is much lower. If we had a tetraploid, there would be 36 chromosomes with 4 sets of 9 chromosomes. We do have an even number of sets here. Chromosomes could pair and divide, and perhaps this plant could be fertile, but we don't have any tetraploids on the market, so it's somewhat irrelevant. The other polyploids, Floritam, Provista, and Citrablue, actually have chromosome numbers that are in between triploid and tetraploid. For instance, Floritam has 32 chromosomes. So if we look at what a tetraploid might look at, and then we remove 4 chromosomes, we have a hypothetical case here for what might be the chromosome makeup in Floritam. We actually don't know. This is just hypothetical. What you see here is that we have 2 sets with 9 and 2 sets that are each missing 2 chromosomes. And you can imagine as we try to form a gamete that's a mosaic of all of these, we may not have a correct gamete form. So there would be low fertility of these as well. So this was a long drawn out explanation to show you why diploids are fertile and these polyploids have reduced fertility. And again, a long drawn out explanation to provide some substantiation for why there may be contamination in sod fields. This happens to be a Floritam stolon, and it's a Floritam sod field that's contaminated with a diploid plant. You can see the finer leaves, shorter internodes, very characteristic of what we would refer to as a dwarf or semi-dwarf. And so that could have been from seed that came over from equipment or seed that was dropped by a bird or other animal, any numerous means. In fact, contamination is really not something that we can completely prevent, but hopefully we could delay using frequent mowing or mowing short enough to remove seed heads, barriers between fields, and cleaning of equipment. In terms of the genetic analysis, Dr. Harmon referred to several samples that were collected in Boynton Beach in early 2020. And these were off of sites where LVN was infecting the landscapes. Most of these were sites that had been replanted and were subsequently failing. We also collected sites where the Floritam had been replanted and the plants were living, or the replaced grass was living and looked well. In all, we had 33 single stolen samples collected from these lawns. Again, some of the samples had LVN, even though it was regrassed with something that was supposed to be resistant to LVN. LVN was present, and some samples were LVN free. All samples we brought back to Gainesville, and my lab coordinator, Jamie Buellman, ran DNA markers on these. All samples with LVN turned out to be Floritam. So even though a different grass was the intended purchase and replacement in the landscape, that ended up not being the case. So, and in fact, in every case where we saw LVN, it turned out to be Floritam. This includes lawns intended to be planted with another cultivar. The frequent mistake that was occurring here was the intended cultivar was supposed to be bitter blue. but turned out to be Floratam. Other things we've done, we've worked with sod growers to try to assess whether or not the virus is present on their farm. 16 farms have voluntarily submitted samples, and we have tested for the presence of sugarcane mosaic virus. And to date, we have not detected any sugarcane mosaic virus on any of the sod farms that have worked with us through this process. I think that should be comforting for consumers out there. And so with cultivars, kind of where are we? So there are these 8 cultivars that are available for purchase in the state. Again, Raleigh is only available in the Panhandle, so it's really not that relevant for LVN discussions, but it needs to be listed. So when it comes to LVN replacement options to date, we're at Seville, Classic, and Palmetto as those options that are out there suitable for replacement of Floratam in LVN affected landscapes. We formerly recommended bitter blue, but because of the frequency of individuals who purchased bitter blue, that subsequent failure, and that ending up being documented that the failed replaced grass was actually Floratam. Because of the frequency of that occurring, we've taken bitter blue off of this replacement list. Maybe it can come back in the future, and perhaps in the future we'll be able to add Provista and Citra Blue to this list. They are both under evaluations in LVN affected sites, but we need more time to be certain about how these new grasses on the market are performing with respect to LVN. With respect to certification, certification is a law-abiding process authorized by the Federal Seed Act, administered by the Association of Official Seed Certification Agencies, AOSCA. In Florida and Alabama, the Southern Seed Certification Association is the recognized seed certification agency. And this gives us 4 classes of certified sod: breeder's sod, foundation sod, registered sod, and certified sod, each coming with their associated colored tag that designates their level. How does this process of certification work? Someone may develop a new cultivar, and then they would create a breeder's field of that. And generally the breeder takes care of that, and then use the breeder's field would be harvested to plant a foundation field. That foundation field, at least when it comes to UF released products, is under the direction of Florida Foundation Seed, which may work with licensed producers. From foundation, that sod would be harvested to plant an expanded registered field with licensed producers. Those licensed producers could then take that registered field and plant a certified field, again, only with licensed producers. So this is a process of ownership through this, and it is legally binding, and there are inspections that occur through this process. So fields are fumigated, or there's an alternative method of fumigation prior to initial planting. The different classes can be planted based on the need or designation based on the SSCA inspection. And inspections are based on isolation, weed pressure, and genetic integrity. Fields must be approved before harvest, and shipment of sod should contain appropriate certificate depending on that class. So where does that leave us with these cultivars that are on the market, and how can we ensure genetic purity? There's nothing in place to oversee the genetic purity of Floratam, Bitter Blue, Seville, or Raleigh. In the case of Palmetto, those programs will be covered later in the webinar. For classic St. Augustine grass, the owners do maintain fumigated fields to ensure genetic purity, although there is no licensing program. For Provista, there is a licensing program, and the fact that it's glyphosate resistant should prevent contamination from other cultivars, ensuring genetic purity. Citra Blue is the only of these that follows the certification protocol. However, there is an important caveat in that production fields or sod sold to consumers are not a certified class, but they are required to have been planted from a certified class of sod. So if we look at the consumer down here, they're going to receive sod from a production field of Citra Blue. That production field must be planted from either foundation, registered, or certified, and it would have that inspection go along with it from Southern Seed Certification Association. So there is a nice chain of command of ensuring genetic purity that goes into any square foot of Citra Blue that would be sold to a consumer. And that's all I have. Thank you.
All right, thank you, Dr. Kenworthy.